Cal State University at San Marcos, the genomic landscape of Neanderthal ancestry in modern humans. Let's see if you can share it.
Okay, so I'm sharing my screen so hopefully everybody can see it. Let me go ahead and get started on here. Well, first off, this is pretty phenomenal, and I'm super nervous right now because there's like 500 something people joining in from all over the place, and that's pretty overwhelming. So hopefully everybody's staying safe out there and sane out there, and keeping great research going. So thank you to all the organizers for doing this phenomenal job of organizing everything together. So I'm going to tell you about some of my recent work that I've been doing in analyzing the 1000 Genomes Project data, as well as looking at Neanderthal tracks or putatively introgressed Neanderthal tracks into modern human populations to understand why exactly we have regions of depleted or enhanced Neanderthal ancestry. Okay, so this is, and this was a phenomenal, very inspiring figure, I should say, from this very cool paper by Renaud and Aki, where they looked at, archaic variation, specifically in this case from Altai Neanderthals in modern human populations, estimating somewhere between 1 to 4% of Neanderthal ancestry across modern human populations. And lots of subsequent papers have looked at possible hypotheses as to why you should have regions of depleted or higher Neanderthal ancestry, specifically looking at East Asian populations putatively having about 12 to 20% of greater Neanderthal ancestry than European populations, potentially because of secondary contact from Neanderthals. And so then, it always sort of, begs explanation. Why would we expect that, right? I mean, if here's a simple cartoon, just kind of looking at sort of what exactly we would expect under, just putatively recombination processes, neutral processes. When you have a human grandparent and a Neanderthal grandparent, they produce these gametes that are recombinant, and then you put those together, you have all these sort of novel mutations that say arise in some of these individuals, and then this proceeds, and then, fast forward a few thousand years, if everything goes just how it's supposed to go, right, you would expect on an average sort of putatively equitable degrees of Neanderthal and human ancestry over timescales. But clearly that's not what's happening, right? And so it begs the question, why are some parts of modern human genomes have more or less parts, more or less degrees of Neanderthal ancestry? And so the way I'm going to address this is by going back to sort of a traditional isolation with migration model, from 2001 by Nielsen and Wakely, and then subsequently wrote into programs by Jody, my postdoc advisor, myself, and Vitor Sousa, into a variety of problems. And so the idea behind this model is pretty simplistic, where you have an ancestral population sometime in the past that diverges at some point t. This ancestral population is of size NA here, and it splits into two populations. And they continue to exchange genes at rates M1 and M2 all the way up until the present. And then you sequence or you sample individuals from the present, and then you try to infer these under this sort of traditional isolation with migration model. And so I'm going to take this approach, but we're going to kick it up a little bit of notch, and we're going to understand two concepts or two tests that we can sort of tie into this isolation with migration model. And the first one is what I'm going to call a test of differential introgression. And the idea here is that let's assume that, say, here is one population sometime in the past, and you have a second population sometime in the past again. Let's say this is human, and that's Neanderthal. And let's say there happens to be a mutation on this background right here, which happens to be disadvantages in this population. And so as they continue to exchange genes, you would expect over time that you would have a depleted degree of ancestry, which carries that allelic variant because it's disadvantageous in this background. Whereas in this population, you would expect that this mutation possibly goes to fixation. And so the idea is that if you estimate the migration rates at these locations, you would expect the migration rates to be completely different between this particular locus compared to the rest of the alleles at the rest of the genome. So by that, what I mean is if I estimate the migration rate at, say, this locus right here, that is going to tell me that the net degree of migration rate is very low compared to the migration rate at this locus where I have this allele right here, which is putatively neutrally evolving. And so it is increased in frequency in this population. Okay, so the idea here is that if you estimate migration rates, you would see differential rates of migration between loci. And so this is commonly, people put this, call these as genomic islands of speciation, because putatively they could contain loci that are highly differentiated between the two environments or two populations, leading to eventual reproductive isolation and eventual speciation, right? And so here's an example, for instance, from this cool paper that was looking at 3-spine sticklebacks understanding, looking at FST, traditional genomic sort of island differentiation analyses, as they call it, looking at FST scans to look at how different are these two populations. If they are very, very different between them, then putatively they are not exchanging genes at high degrees between those populations at that locus. And so here they identify these as genomic islands, right? So that's the idea behind this first test. And so the way I'm going to address this is by calling this as a test. It's a likelihood ratio test of differential introgression, where under a traditional IM model, if the null hypothesis here is that, and I estimate the migration rates across a set of loci, I would expect to observe only one group of loci where the posterior density distribution of this migration rate estimate would look unimodal in this case across all loci. On the other hand, if indeed there happens to be an alternate hypothesis that is true, in that there are two groups of loci where there are one group of loci that are exchanging genes at a or exchanging alleles at a lower rate, and you are having a second group of loci that are exchanging genes or alleles at a higher rate, you would see a bimodal distribution. And indeed, you can classify these loci as having lower migration rates and having higher migration rates. And so this test is what I'm going to call as a test of differential introgression. The second idea that I'm going to talk about is a test of linked natural selection effects. And so here is an example where, let's say I happen to have an advantageous mutation that shows up in a population. Eventually you would notice that over time, let's say after 10 generations, the loci that are sitting right next to it, and by virtue of the fact that these are linked or they're sitting really close to each other, they would recombine together, and you would see that these haplotype tracts would increase in frequency within a few generations. And so let's say fast forward 100 generations, you're expecting to see a greater degree of linked natural selection at this locus. In other words, what's happening at these loci is that the net diversity at these genes or these loci would decrease, whereas everywhere else, the net diversity, if they're putatively evolving neutrally, would just, would be higher. Okay, so we're gonna do a test of, and these are seen again, right? This was another cool paper that was in 2014 where they looked for linked natural selection effects across populations of European crows, and they looked to see that a bunch of these linked genes had reduced diversity, and as it turned out, a lot of these happened to be visual perception or pigmentation genes in European crows, right? So here I'm proposing to do a second test, which I'm going to call a test of linked selection, where if I estimate the population sizes or effective population sizes as theta under an IM model, if indeed there's only one group of loci where nothing else is happening, everything is evolving at the same same demographic model, then I would expect to see a unimodal distribution for the estimated population sizes. On the other hand, if indeed there are linked selection effects, then I would expect to see reduced diversity or theta at some of these loci, whereas some of the other loci where there are no linked selection effects, I would expect to see a second mode, which I'm going to call as an alternate hypothesis here. And so, so here's the model that I'm trying to test here. So I'm going to look at Neanderthals and Europeans under this sort of, and Neanderthals versus Asian populations under this traditional IM model, and test for the effects of linked natural selection and differential introgression. And we've done this, we wrote a nice piece of software called MigSelect. There's a preprint out there. I'm happy to answer more questions at a later point, where we basically classify each locus as having high versus low migration rates or high versus low diversity or population sizes. And then we use a likelihood ratio test, and then use base factors to provide support or evidence for one hypothesis versus the other. Okay. So the idea here is that if it's close to 0, there's no evidence for either hypothesis. Greater than 1, in this case, has evidence for the alternate hypothesis, which indeed that there are 2 groups of loci, either for migration rates or linked selection effects, because there's 2 groups of loci having differential diversity. Or if it's less than 1, there's evidence. There's evidence for the null hypothesis, which that all the loci are indeed evolving under the same demographic model. And so here's what we did. So we obtained about 150 to 200 genomic regions, 5 kilobases upstream and downstream of regions of putative Neanderthal ancestry. Matthias, I wrote to Matthias from this really cool paper in 2018 in Molecular Ecology, where they used this method to estimate regions of putative Neanderthal ancestry. And so then my grad student, Melissa, and a couple of other folks helped me sort of sift through the 1000 Genome Project data and look for regions that are upstream and downstream of these putative regions. And we were able to collect intact about 150 to 200 loci, depending on the model. From the 1000 Genome Project's data from about 20 unrelated individuals. So that's about 40 genomes from across CEU and CHS and CHB. And we ran MigSelect to identify loci under linked selection effects versus differential introgression effects. So I'm going to run you through some of the results that we found. So these are demographic estimates under the traditional IM model. And these sort of broadly agree with what's been previously estimated for these populations already. Remember that, so these are again scaled population sizes. So these are theta estimates. So theta is 4 any mu, and then T is scaled by a mutation rate as well. So this is divergence time divided by the per site mutation rate. And then the migration rates I've shown you overall is just high or low, because the confidence intervals around these were higher, because we included regions both that were introgressed putatively as well as regions that were not introgressed. And so these were, but the overall signatures look to be the same, but there seems to be higher migration rates from the European populations into Altai, or from CHS or CHB into Altai Neanderthals. The overall size of the Altai Neanderthal population sizes is much smaller compared to the Europeans or the CHS, CHB populations. And the ancestral populations are much, much larger, indicative of the bottleneck event due to out of Africa. And it also shows you that the divergence times in this case from Altai and CEU seems to be a little lower than the Altai and CHS and CHB. So these are sort of things that, again, they have confidence intervals, and so these sort of agree broadly with what we already know. But here's the cool stuff. So when we did these likelihood ratio tests of linked selection and differential introgression, oh, bam, 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 right? Unfortunately, my p-values were, pretty high here, and I was kind of bummed about it. But regardless, so what we broadly found was that there seemed to be no linked selection effects or differential introgression effects across each of these population pairs. Whether you compare CEU and Neanderthals or CHS and Neanderthals, the p-values in these tests all indicate that they all support the null hypothesis, in that there's only one single group of loci. But I saw this one right here, right? And I was getting a little hopeful here that this one's a little closer to what I would expect. And so I went ahead and I looked at exactly what those loci are and the distribution of base factors for these loci for linked selection effects in CEU and Neanderthals. And so here's what that looks like. So here I'm orienting you, I'm showing you base factors, in this case log scale, on the so on the y-axis. The x-axis are just the 138 loci that we looked at, and these are loci that are enhanced in this case for lower diversity. And there's a handful of loci that kind of stick out for having a higher support for having lower diversity at these loci. On the other hand, if I look at migration rate estimates, all of them seem to support the fact that they all fall under just one class of loci. Now, if I look at the same thing across CHS, CHB, on the other hand, there's none of these loci that seem to stick out as having either low theta or low or high migration rates between the two. So these are my results from CHS and CHB. So then we went ahead and we looked at what exactly those handful of loci that stuck out as having linked natural selection effects in CEU. I went ahead and I looked at exactly what's sitting in those loci. And as it turns out, there were at least a couple of genes that we were able to pull out. There was this MUSK and GKAP1 gene, as well as a bunch of line and sign elements, which were also involved in this. So some promising stuff. In summary, linked selection effects may have caused increased Neanderthal ancestry at some neighboring genes in Central Europeans. and no detectable differential introgression. So we want to analyze more populations, more archaic populations, and understand sort of functional consequences for enhanced Neanderthal ancestry from here. So with that, I'd like to thank NIH and NSF for funding, a bunch of folks, Jody, Melissa, Andrew, and Jared, who've helped me with a lot of stuff. We also have the software on my Git page, and I'm happy to answer any questions if we have any time. So, thank you.
Thank you so much. We sort of out of time, but maybe there are two questions you can answer in Q&A later. Maybe I'll just ask the first one from Kelly Harris. What happens when the migration rates and vector population sizes are over-dispersed across loci rather than bimodal?
That's a great question. Unfortunately, right, if they're broadly dispersed, then there's no sort of statistical power for us to actually look to see which one is higher or which one's lower. So yes, it does not have the power to actually look at that. So my guess is that that's possibly what is happening. In fact, when I looked at the posterior density distributions across migration rates, for instance, they were not unimodal or bimodal at all. They, sometimes they tended to have sort of like flat distributions. And so, my guess is also that I would have to probably look at more loci to be able to sort of decipher that a little better.
Thank you so much. That's a great session. Wonderful start to PEQG. We are concluding now. The next session is at 3:45 Eastern Daytime. The evolution of gene expression is going to be chaired by Daniel Matute, and hope to see you very soon.